There are different types of nucleic acids. Nucleic acids are an important class of macromolecules found in all cells and most viruses. The functions of some nucleic acids have to do with the storage and expression of genetic information. Nucleic acids are generally very large molecules. Indeed, DNA molecules are probably the largest individual molecules known. There are a few other simple yet important types of nucleic acids. In this video, we will study those simple individual nucleic acid derivatives such as ATP, cyclic AMP, NAD, and FAD. One of the main nucleic acids, the DNA, which encodes the information the cell needs to make proteins, is one of the most important nucleic acids that we know. A related type of nucleic acid called RNA comes in different molecular forms that participate in protein synthesis. We will study DNA and RNA in separate video lectures. Now, let's take a look at a special group of molecules that are nucleic acid derivatives. Nucleic acids are made up of building blocks known as nucleotides. The nucleotides combine with each other to form polynucleotides such as DNA and RNA. But these two are not the only type of nucleic acids. There are a few simple ones as well. One of them is ATP, that is a nucleoside triphosphate that transports energy within cells for metabolism. Another one is cyclic AMP that facilitates and promotes the mobilization of glucose and fatty acid reserves. Nicotinamide adenine dinucleotide, or NAD, is a signaling molecule as well as a cofactor or substrate for many enzymes. It plays a significant role in metabolism. Finally, FAD, similar to NAD, delivers hydrogens and electrons to biochemical processes that can use the electrons and hydrogens to make ATP. Both NAD and FAD are responsible for accepting high energy electrons that are used to synthesize ATP molecules. Adenosine triphosphate, or ATP, is an organic molecule that provides energy to drive many processes in living cells. Cells require chemical energy for three general types of tasks: to drive metabolic reactions that would not occur automatically, to transport needed substances across membranes, and to do mechanical work such as moving muscles. ATP captures chemical energy obtained from the breakdown of food molecules and releases it to fuel other cellular processes. Found in all forms of life, ATP is often referred to as the molecular unit of currency of intracellular energy transfer. From the perspective of biochemistry, ATP is a nucleotide that consists of three main structures: a chain of three phosphate groups, the sugar ribose, and the nitrogenous base adenine. In the context of biochemical reactions, the phosphate to oxygen to phosphate bonds are frequently referred to as high energy bonds. I will talk more about this in a moment. ATP is not a storage molecule for chemical energy. That is the job of carbohydrates such as glycogen and fats. When energy is needed by the cell, it is converted from storage molecules into ATP. ATP then serves as a shuttle, delivering energy to places within the cell where energy-consuming activities are taking place. Like I mentioned earlier, the energy is stored in chemical bonds connecting the phosphate groups. This means that the phosphate tail of ATP is the actual power source. Available energy is contained in the bonds between the phosphates and is released when they are broken. Usually, only the outer phosphate is removed from ATP to yield energy. When this occurs, ATP is converted to adenosine diphosphate, or ADP. ATP is able to power cellular processes by transferring a phosphate group to another molecule. This transfer is carried out by special enzymes that couple the release of energy from ATP to cellular activities that require energy. The structure of ATP has an ordered carbon compound as a backbone, but the part that is really critical is the phosphorus part, the triphosphate. Three phosphate groups are connected by oxygen to each other. And there are also side oxygens. Connected to the phosphorus atoms. Under the normal conditions in the body, each of these oxygens has a negative charge and therefore repels each other. These negative charges want to escape to get away from each other. So there is a lot of potential energy there. If you remove just one of these phosphate groups from the end so that there are just two phosphate groups, the molecule is much happier. Just the cutting of one bond with the accompanying rearrangement is sufficient to liberate about 7.3 kilocalories per mole of ATP. This is about the same as the energy in a single peanut. Cyclic AMP is a second messenger important in many biochemical processes. The cyclic AMP is a derivative of adenosine triphosphate, or ATP, and is used for intracellular signal transduction in many different organisms. Cyclic AMP is synthesized from ATP by the enzyme adenylate cyclase located on the inner side of the cell membrane. The cyclic AMP is involved in the regulation of glycogen, sugar, lipid metabolism. The cyclic AMP is used in intracellular signal transduction, like I mentioned before, such as transferring into cells the effects of hormones like glucagon and adrenaline, which cannot pass through the cell membrane. It is also involved in the activations of chemicals called protein kinases that eventually target the cell's DNA. In a signaling pathway, cyclic AMP enzymes and activated proteins interact with specific proteins that eventually lead to a change in the cell's environment. The chief role of cyclic AMP in several tissues is to facilitate or promote the mobilization of glucose and fatty acid reserves. Nicotinamide adenine dinucleotide, or NAD, is found in all living cells. It is called a dinucleotide because it consists of two nucleotides joined through their phosphate groups. That is why it belongs to the nucleic acid family. In metabolism, NAD is involved in redox reactions, carrying electrons from one reaction to another. These electron transfer reactions are the main function of NAD. However, it is also used in other cellular processes, most notably as a substrate of enzyme in adding or removing chemical groups to or from proteins. Because of the importance of these functions, the enzymes involved in NAD metabolism are targets for drug discovery. In biochemistry, flavinamine adenine dinucleotide, or FAD, is a coenzyme associated with various proteins, which is involved with several enzymatic reactions in metabolism. In metabolism, FAD is involved in redox reactions, acting as an electron carrier. FAD also has non-catalytic functions, whereby it is involved in the blue-sensitive light photoreceptors that regulate biological clocks and the generation of light in the bioluminescent bacteria. FAD forms certain flavoproteins when joined with certain proteins. These proteins are essential in many biological processes such as DNA repair, photosynthesis, and the removal of free radicals. Thanks for watching.